Good evening. Today is week 11th session, and in this session, we'll be covering the week 11 topics such as biological network analysis, mutations and signaling, and pathway enrichment. So, the MCQs will be related to these topics, and along with MCQs, we'll also look into certain concepts and certain topics which will be in those MCQs. Along with that, we'll also see the current literature, that where is it going and are there any updates about it. And if you have any doubts regarding the assignment, so that will also look into. So let's start. Okay, so I'll start with the first question. So first question is, what is a biological network? So options are option A, a database of biological sequences. Option B, a graphical representation of biological entities and their interactions. Option C, a protein structure. And option D, a cellular organelle. So now, yeah, this question is quite straightforward and easy also, that biological network is a graphical representation of biological entities and their interactions. So it is not just the database of sequences, and it's not a protein structure, and it's not any organelle. So let's look into the other common types of biological networks. So the other common type of biological networks are protein-protein interaction networks, metabolic networks, genetic interaction networks, gene and transcriptional regulatory networks, and cell signaling networks. So each and every network, uh, as they all include the biological entities, so that's why they comes under biological networks. And second thing is that whenever you see their name, so from their name only, they hints that what will be their nodes and what will be their edges. So nodes are like these entities, which could be either proteins, metabolites, and these arrows are the edges. Edges means what is the relationship between those entities. So for example, if this square box represents the metabolites and the circular box represents the protein, and this arrow represents metabolism. That means these proteins and this metabolite, there is some metabolism undergoing which is converting this protein into a metabolite. So this is a simplistic representation of a network in which they have combined different type of biological entities. So this is an overview of biological networks. In this, they have not restricted it to a single kind of a network, but it is just the entire representation of all the entities and what could be their possible relationships or edges. So now let's move to next question. So next question is, in a protein-protein interaction network, what do nodes represent? Option A, DNA, option B, genes, option C, proteins, and option D, pathways. So as we have discussed that what are the nodes? Yeah, so you are right. The correct answer is option C, that is proteins. Now we'll look into the protein-protein interaction networks. So for detail, you can go through this link because at EBI they have clearly explained all kind of networks and they've also given the figures. So it will be easier for you to understand from there if this is not clear from here then. So protein-protein interaction networks, they represent that different proteins are connected to one another and how they're interacting with one another. So to visually see them, we generally opt for the protein-protein interaction networks because we can visually see them. Otherwise, reading through the paper or going through the tables is quite complicated. So that's why this interaction networks provide you the easy representation. That's why we opt for them. And you can also understand that how some proteins are more important and they are more interacting with multiple partners. So in that way, that protein is more significant, or you can say you can study that protein if it makes many interactions. So in that case, it protein-protein interactions can also tell you that what is the physiology of the cell. Either it is a normal state or disease state. If you know what are the interactions the protein is supposed to make, and whenever you will be comparing in both the normal state and disease state, so there will be some change in their interactions. So with that, you can also predict that what is the cell physiology, whether it is a normal cell or it is a disease state. So in this way, interactions can also help you to identify the physiology of the cell. And along with this, it will also help you to design or develop the drugs, since drugs can also affect the PPIs. So there are certain protein-protein interactions which you might see in the cancer phenotype, for example. So whenever you design some drug, so what you are doing with that drug, you are allowing your protein to interact with that drug so that your protein does not interact with the other protein. So in that case, it will also help you to observe that whether your drug is working fine or not. So these protein-protein interactions in this way, they help in drug development as well. And the protein-protein interaction networks are the representation of these PPIs. So many people, they call PPI as a network, but otherwise the complete term is PPIN. That means protein-protein interaction network. So it is a mathematical representation of physical contacts. So as we have seen in the gene regulatory networks, so in that case, in gene regulatory networks, there is no physical interaction between two genes. There are some regulations that one gene is stimulating the expression of other genes through promoters or any other factors. So in that case, there is no such physical interaction between two genes. But in the protein-protein interactions, the interactions are physical interactions where two proteins are interacting with one another from some site. So these contexts, they are quite specific. Specific means that there are certain sites which are specific for that interaction or binding. Not all the site of the proteins will interact. So that's why these contexts are quite specific and not each and every protein will bind to any random protein. So that's why also these contexts are specific. They occur between defined binding regions in the proteins. So as we discussed that there are certain regions, interfaces where the interactions will happen. And the other thing is that they have a particular biological meaning. So that interactions have some purpose. Either it helps in the stability of the protein or it helps to produce or catalyze some other reaction or means there is some function which is associated with those interactions. And these interactions can either be stable interactions or they could be transient interactions. So when we say stable interactions, it means that whenever two proteins will join, so it will be stable. It remains fixed for some time. It will develop a complex, as we have seen in the case of ribosomes. So during the protein synthesis, during translation, we see that there are two subunits of ribosomes. So those two subunits come together and they join. So that interaction is stable interaction. Whereas if we talk about the transient interactions, that means they are very brief. They are for a very short period of time and that modify or carry a protein, which is leading to a further change. So for example, if we talk about the protein kinases or nuclear pore importance, so these proteins, they just do transient interaction and they allow the protein to move or they are means adding phosphate group to some other protein. So these are very quick interactions. So they constitute the most dynamic part of the interactome because they are continuously changing. They're not fixed or they're not remaining for a certain longer period of time. And now let's see that what are the advantages of these PPI networks, what we can learn from them. So first thing is that they assign putative roles to uncharacterized proteins. For example, if there is some protein which is not characterized, so by looking at its interactions, we can understand that this could be the role of this particular protein. So for example, protein A is interacting with the kinases family proteins. So with that, we can say that this particular protein might have some role related to kinases or means with that we can kind of assign it some potential role and then add fine-grained detail about the steps within a signaling pathway. So with interactions, we can also predict that which step this particular protein is involved in. So there are multiple reactions which are going in a pathway. So with that, we can also characterize that what exact reaction step this particular protein could be involved in, then characterize the relationships between proteins that form multi-molecular complexes such as proteosomes. So these interactions also we can understand because there are multiple proteins in our body which exist as the different subunits. And then during a reaction or during a pathway, they all subunits get combined, such as proteasome or such as spliceosome during splicing multiple spliceosomal complexes. So multiple subunits come together and join and then it leads to splicing. So similarly, there are other examples as well. So in that way, these are the advantages of protein-protein interaction networks. Then comes the interactome. So before moving further, is it clear till now about protein-protein interactions? Okay, so now let's see that why this interactome is important because these protein-protein interactions, they are interacting, these proteins are interacting with one another. So we can call it as an interactome. That means the complete study or the totality of the protein-protein interactions which can happen in a cell. So from interactions, the term interactome has come. So we have seen proteome, genome, then other terms, transcriptome. So similarly, interactomes is total of all interactions. And there are development of large-scale PPI screening techniques which are capable of identifying such interactions, such as mass spectrometry can also help you to identify the protein-protein interactions as well as these two hybrid assays. So these are some of the techniques with which people try to look into the protein-protein interaction networks experimentally, that what are the possibilities that two proteins are interacting with one another. So in that way, this is the representation of interactome. So the left side represents the yeast. So there are multiple proteins in the yeast which are interacting, and the right side represents the human interactome, in which there are multiple human proteins which are interacting to one another. So these darker lines in the middle, they are the edges. So with which we can, if we have to compare both of them, so we can say in the case of human interactome, it is well connected. More and more edges you can see, as there are multiple red and blue edges here. And on the other end, we can also say that there are multiple proteins which are involved in the interactions in case of human. That's why more edges are also observed as compared to the yeast. And these interactions were captured by a yeast to hybrid method. So yeast to hybrid method is a method with which we can experimentally identify such protein-protein interactions. And now I'll talk about some of the properties. Okay. Yeah, so in this case, when we talk about the mass spectrometry and protein-protein interactions, so we cannot directly say that just mass spectrometry, we have to combine some other methods such as high throughput affinity purification. So after affinity purification, we go for the mass spectrometry. So for example, in a sample, there are multiple proteins which are present. So you will allow certain complexes only to be present in that sample, and you'll remove the, for example, means there are multiple screening methods with which you can screen the monomeric proteins, or you can say the proteins which are not forming the complexes. So in that case, you can simply filter out all those proteins and you will only retain the complex. So after that, if you will perform mass spectrometry, so you will only observe the peptides which will be coming from the proteins which are involved in the complex formation. So that could be one of the method with which you can understand that these proteins are forming a complex. That means they are interacting with one another. So that is one kind of way with which mass spectrometry helps you to identify the protein-protein interactions. But yeah, it's not directly helping you, but there are other purification methods with which you can use the mass spectrometry. So it provides you the evidence at the level of protein-protein, I mean sequence level. So okay, yeah. Now we'll talk about the properties of these protein-protein interaction networks. So these properties are in general, these are the properties for most of the networks, not just biological, but all other networks also follow these properties. But in this case, as we are talking about PPI, so I've written as properties of PPINs. So first is the small word effect. So as the name say, small word. So that this is the term which was taken because in the previous time there was one experiment of the passing the parcel kind of experiment, in which it was observed that this word is very small. In maximum, at maximum, 6 people are required to transfer any parcel from one place to another. So with which, after studying all of these, it was observed that uh, if you want to reach from one node to the another node anywhere in the network, so at max you have to move through the 4 intermediate nodes. So there are total 6 degrees of separations, or 6 network means 6 contacts or 6 nodes will be observed in any information passage. So this is just the average concept. So after performing multiple analysis, average was calculated. So that's why the name small word was given, that there are very lesser difference between 2 nodes which are lying far away from one another in the network. So which means that there is great connectivity. That's why the word is small. So in other words, it can be said that network's diameter. So when we say diameter of a network, it means that maximum number of steps which are separating 2 nodes. So there are multiple nodes in the network. So we will select those 2 nodes. And in this case, the steps will calculate only those steps which are the minimum number of steps, or we can say the shortest path. So, for example, if I have to reach from A to B, so this is my shortest path. Otherwise, I can also go through this, and again going through means here it is no way. So, for example, I go through this, and then I go to this, I go to this, and if I take these 2 nodes also instead of going directly to B, so in that case, this is also one possible path to go from A to B. But what we do in the protein-protein interaction or any other network, whenever we are going to find that how we have to reach from one node to another node, so we will only take the shortest path. So what we will do for all the nodes, we will calculate all the shortest paths, and the path which will be maximum among those shortest paths will be referred to as the diameter of the network. So it is the maximum steps. which separate any two nodes. So if there is a small word effect, it means that network diameter will be small, no matter how big the network is. So that's why this small word name was given, and it means that two nodes are separated by less than 6 steps. So there is also another term which is linked to small word effect, which is 6 degrees of separation. So degrees means how many edges or means how many connections are involved. So that's why 6 degrees of separation. That means two nodes are separated by the 6 degrees. So that is the max degrees with which I can reach from one node to another node, which is lying anywhere in the network, farthest from my given node. So this is one of the property, that is small word effect. Now the question comes that this level of connectivity is very important, and it is also showing that the network is efficient and there is quick flow of signals within the network because the separation is less. But then the question arises that if network is so tightly connected, that means if there are very few number of steps it has to take, then why don't perturbations in a single gene or a protein have dramatic consequences for the network? So for example, if any of the gene in the middle, if it gets destabilized, or if it loses its function, then why not my network get disturbed? So this question was raised, and then new property came up. Then it was observed that these protein-protein interaction networks are also scale-free networks, in which majority of nodes in the scale-free networks have only fewer connections to other nodes. So not each and every node is connected to multiple other nodes, but there are few nodes. So these few nodes are kind of helping you to reach from one place to another place with the shortest steps, shorter number of steps. So that's why it is making sure that even if some genes get loses their function or loses the interactions, or for example if some protein loses its interaction, so in that case also my whole network is not destroyed. So with that they have said that there are certain nodes which have major degrees, that higher degree values, and there will be very few means very few number of nodes will be there which will have the maximum degrees, and majority of nodes will have lesser number of degrees, that means lesser number of connections. And the nodes which have the greatest number of degrees or greatest connections, they are also called as hubs. So this is a graph which represents the scale-free network degree distribution. So degree distributions, degree is meaning connections, number of connections, and the y-axis represented number of nodes. That means how many proteins or how many genes are involved. So in this case, you can see that majorly the genes, it is going downwards as we are increasing the degrees. So which states that majorly the nodes have the lesser degrees, but there are few nodes which have maximum degree values. So that's why these nodes are hubs, and network is scale free. That means some have lesser connections and some have greater number of connections. Yeah, so we have already discussed it, that degree is the number of connections and the scale free network. So this kind of distribution is also known as power law relationship. So which means few higher degree nodes and many low degree nodes. And then there is also another mechanism. So as we know that these protein-protein interaction networks, they're not always stable. They're dynamic interaction networks. So they keep on changing their interactions. So in that case, whenever we have to decide or the attachments or the interactions that will happen, so there is a model which is known as preferential attachment model. It is also known as rich get richer. That means a node which is connected to multiple several nodes. So there are more chances that it will get attached to new node if new node is entered into a network. So that is referred to as preferential attachment model, which suggests that new edges are more likely to connect to nodes with higher degrees and which facilitate the self-organization and growth of the network. So in general, what happens is that whenever you are studying some protein-protein interaction network, so what people does that whenever they give a new protein or the new node, so then we have to decide that to which particular protein it will interact. So in that case, preferential attachment model is considered. So it is most likely to get connected to a hub. That means which is already connected to multiple interaction partners. So this is just a model or algorithm which is followed whenever a network growth is observed. So network growth means whenever you are adding more and more components to it to increase its size. So in that case, to understand or identify the edges, you follow this algorithm, which is preferential attachment, in which what you do is that you look for the nodes which have maximum number of connections and you assign that new protein which is added to the network to that particular node. So that interaction will be added. This is known as rich get richer algorithm. So the third property of protein-protein interaction network is transitivity. So transitivity is also called as modularity, that how modular your protein is. So means how much nodes are clustering together. So clustering means they are grouping together. So if transitivity is high, that means my network has the nodes which have tendency to cluster together and which can also form the densely connected internally. So, for example, if I have to take an analogy in social science, then we can say that if the friends of my friends are my friends, that means if I have a friend and my friend is having multiple friends, so those friends will also be my friends. So, in that way, this is referred to as transitivity. And this is not always the case, but this is just an analogy. So, most likely it happens that if my friend has some other friends, so they are friend to me as well. So in biological network also, these community identification is very important because they reflect the functional modules and protein complexes. So they will also help you to understand that which are the proteins which might be involved in the interactions. Because why we are studying these interactions? Because there are certain interactions which are having some functional role, which could either be leading to a normal state or a disease state. So we can target those interactions. That's why the understanding of these interactions is important. Understanding that which are the proteins involved in this particular uh, physiology or this particular state is important. So with transitivity, we can identify that. And this is the example. So from the string, it provides you multiple protein-protein interactions. So this is taken from the string and it is representing a module. So there are multiple short modules which are present. So there are multiple groups of interacting proteins, but this particular module is highly connected. So they are self-contained components. That means self-contained means in between them only there are multiple connections that are undergoing. So they are contained in themselves. So well-defined interfaces with other components. That's why they are known as self-contained components. And the defining feature of a module is that its intrinsic functional properties do not change when it is placed in a different context. So these interactions remain constant even if we change the context. And these modules, they also helps to reduce the complexity of biological networks by providing the set of reducible and functional units that can be studied as an integrated entity. So instead of studying all these proteins separately, if we know this particular cluster, so we can simply name it as a cluster A. And we can distinguish them as cluster 1, cluster 2, and cluster 3. And we can study these clusters separately instead of looking to each single node of them. So that's why these modular informations that what proteins are involved in formation of modules is important for us. So that's why that is one of the advantage. So in protein-protein interaction networks, you observe this feature, that is transitivity, that multiple proteins they form clusters. So that is one of the advantage or one of the property. So these are some of the properties of the protein-protein interaction networks. So is it clear till now? Yeah. And this small word or the scale free, so these properties were observed for all networks, such as social networks or biological networks and other networks as well. Now let's look into the question number 3. So in the beginning, I have kept the simpler questions so that the concept built up could be done. So the third question is, which of the following is a common database for biological network information? So option A is NCBI, option B is KEGG, option C is string, and option D is BLAST. Yeah. So the right answer is string because it provides you the biological network information such as protein-protein interaction network. So that information you can get from this database. Now let's look into the homepage of the string database. So you can click on this link and you will be taken to the string homepage. So in this case, there are multiple options which are available which you can explore. So first of all, protein by name. For example, you have a protein name and you want to check that what are the binding partners of this protein present in the organism. So in this case, there are multiple organism information will be present. So for example, if I am interested in human, then I can simply select the human organism or otherwise I can give a protein name. So in this scenario, either I can give the ID, protein ID, or a simple I'll write the protein name. So in that case, it won't be able to auto detect. But if you provide the ID of that particular protein which it takes, so in that case, it will automatically detect. Because for each species, the IDs vary among themselves. So that is one of the way with which it detect the organism. Otherwise, you can select organism and you will click on search. So for that particular protein, it will provide you all the possible interactions. And now one more thing which I want to add is that the string has some algorithm with which it identifies that what are the possible interacting partners of this particular protein is. So first is experimental evidences. So whenever some people are working or experimenting, so they are listing their interactions in some literature or on their servers. So string will collect that information from the publications. The first thing is the experimental evidences. Second thing is theoretically. So means all the theoretical possibilities, what are the possibility of interaction, that also it will judge. And one more thing which it involves is that in the literature, whenever someone publishes some work, so whenever there are two proteins which are appearing more frequently in some literature, so that it provides score to that also. So if two proteins are always observed together in multiple publications, so somehow it gives hint that these protein might be interacting with one another. So this is not always the case, but this is the algorithm. So string uses that algorithm as well. So it is not just based on experimental studies, but other in silico analysis also, theoretical analysis also. So that is one of the way how string captures the protein-protein interactions. So whenever you will provide the protein name, it will look for the other interacting proteins and it will design a network for you. So other options are multiple proteins. So you can also provide multiple proteins and check that to which other proteins they interact. So there are possibilities that these multiple proteins are sharing some of the binding partners. So that is also one of the advantage of studying the multiple protein interaction network. Then protein by sequences. So you can also provide the sequence and then it will compare your sequence to the protein and then identify the interacting partners which are more likely to interact with the protein. Then proteins with values and ranks. Protein families also you can look for pathways, process disease. So this information also means if you have a pathway and you want to know all the proteins involved, so that also you can select from here, or similarly for the process and disease as well. And then add organism. So you can also add the organism, and if you have some interaction data, so you can also add it, or you can be specific. For example, for a specific organism you want to look for all the interactions, that also you can do. Then examples, for example, if you do not have any information and you simply want to just look that what string provides, so you can also go to examples and they'll just tell you. And then comes random entry. So it will randomly give some entry to the protein name so that you can explore their tool and you can understand that how it provides the data. So I have selected random entry because we do not have any. So if you click on the random entry, so in this case, it has randomly taken some protein into consideration. So this is the network which it is providing. So what information it will give you? So it will tell you the nodes which are involved. So network nodes, it has given a definition that what the network node represents in the proteins. So they have mentioned that splice isoforms or post-translational modifications are collapsed. That is, each node represents all the proteins produced by a single protein coding gene locus. So there are, as we know that there are multiple proteins which are possible from a gene. So in that case, it collapse it and take a single protein to represent that gene. So in protein-protein interaction network, they are considering a single reference protein, not multiple isoforms. And they are not taken twice or thrice. If there are three isoforms for one gene, then not three isoforms will be taken into consideration. So one more thing which you will means one more challenge in protein-protein interaction network is that there are certain genes which could give rise to three different proteins. And in that case, those three different proteins can change their binding interaction partners. So some isoform may not interact with the protein with which other isoforms interact. So that is one of the complexity which is involved in with these PPIs. So that's why we require the experimental evidences for that. That's why more and more experimental studies are required for validating these PPI networks. And then comes node color. So they are showing that what does these colors represents. So the colored nodes, the query protein and the first shell of the interactors, and the white nodes represent the second shell of the interactors. So in this case, we do not see any white node, which would be the second shell. Similarly, node content empty nodes, proteins of unknown 3D structure, and filled nodes represent that there is some 3D structure which is available. So there are many proteins which could not be structured in the PDB, or it means which have not been structured yet. So in that case, they might not include this particular uh, filled circle. It will be empty. So in this case, these all are empty, which means their 3D structures are not available. And they're also telling you that what these edges represents. So these are the known interactors. So if the edges are blue in color, which means they are taken from curated databases. So curated databases means the databases which theoretically look for the interactions. So which I mentioned earlier, that if the two proteins are appearing more and more frequently together in the literature, so it is expected that these two proteins are linked to one another or they are interacting. So that means they are taken from curated databases which are performing curations. So curated databases involves both theoretical and experimental evidences and then predicts. So these are kind of predicted protein-protein interactions, whereas the pink lines represent experimentally determined interactions. So if there is experimental evidence such as these two hybrid systems are used or affinity chromatography with followed by mass spectrometry, such things are involved. So which means they are experimentally determined. So these edges, they will represent, these colors represent that what is the evidence for that interaction. And in this case, predicted interactions. So the green nodes represent the gene neighborhoods, gene fusions, or gene co-occurrences. So in this is a kind of a legend with which you can understand that what each and every component of the network means. So in this case, as I have selected random, so they are telling me that what was the input, random input which was taken. If we provide some protein name, so that will be our input. Now it is telling that what are the predicted functional partners. So our aim here was to look for the functional partners or binding partners of my query protein. So this is the list of those binding partners. So in this case, it is a very small network. But if you are looking for some binding partners, for example, if your protein is very highly interactive, so in that case, the list would be long. But in this case, list is short. And they have mentioned that these all are the hypothetical proteins. So they are derived by automated computation analysis, and they are, means because random entry was selected, that's why these all proteins are hypothetical and their structures are also not available. And they have also selected the organism to be Mycobacterium intermedium. So means this was a random entry, so everything they have selected themselves. So otherwise, whatever organism you select, that name will be given. NCBI taxonomy ID is given, and what are the other names for that particular gene, so that is given here. So in this way, you can use string to look for the binding partners or functional interaction partners of particular protein for which you are looking for. Now, if you click on analysis, so firstly, we were looking at the legend. This entire thing is a legend. We can also go to analysis if we want to analyze it further. So this will give me a brief summary of my entire analysis. So summary means what are the statistics. So first thing, how many nodes are there in the network? How many edges are there? What is the average node degree? So average node degree means for each and every node, there will be some number of connections which are present. So if I take the average of all those connections, that will be my average degree. And why we are looking for average degree? Because it tells us that means on an average, what are the connectivity of each and every component present in the network. So if that means if connectivity is quite high, which means all the proteins which are involved, they are means more, there are more chances that they are forming clusters. So it kind of point towards clustering also. And then there is another measure which is average local clustering coefficient. So this is also another parameter which tells that how much interactions are present in the given proteins. So if there are more connections between each and every nodes, if their average node degree is high, so there are chances that its clustering coefficient will also be high. And one more thing which I want to add, which might complicate you. So let me know if you do not understand it. So whenever we take average or mean of something, so there are chances that if one node has multiple connections, so whenever you will take its average, my degree will or my I mean value will get shifted because of that single node which is having a higher degree. So that is one of the problem, that average sometimes interferes if there are some hubs which are present, so that will ruin your analysis. So in that case, average local clustering coefficient is separately collected, means separately calculated for each and every node, and we cannot rely on average node degree for that case. But for example, if I do not have that much outlier in my in that data set, so in that case, average node degree can provide me an idea that my components present in the network are highly connected. So that means they are interacting with one another more and more. Then expected number of edges. So this is telling me number of edges which are expected for each and every node. And then PPI enrichment p-value. So this tells that what is the significance level of that enrichment of the interactions. So in this case, because whenever you are looking for your proteins, so there will be some hypothesis which you are proposing that this particular protein is involved in the cancer pathway and you are designing drug for that. So if you want to comment that my particular protein was involved in this pathway, it was interacting with these proteins, and now by using this drug, these interactions are inhibited and it is no longer interacting with these proteins. So in such cases, the PPI enrichment p-value becomes important because you should have some significance associated to your analysis. Or for example, if two proteins are interacting, so you also need to have confidence that whether they are interacting, they are strong interactors or not. Because if you are completely based on theoretical predictions, so in that case you cannot completely prove your hypothesis that these two proteins are interacting. So such cases p-value is helpful, which tells you that whether the interactions are enriched or not enriched. So in this case, they have written below that your network does not have significantly more interactions than expected. So there is some expected interactions which are expected for a protein-protein interaction network to have. So this particular network does not have significant number of interactions. So that is seen by the p-value as well. Now they have written local network cluster, which is the string, and in this they have mentioned the cluster ID. So because if multiple proteins are interacting and if all those nodes are interacting with one another, then they will form a cluster. So in this case, as there are only 4 nodes, so I only observed one cluster. But if the network is huge, then in that case, multiple clusters could be observed. So they have written the cluster ID and this brief description of that particular cluster, that in which particular path or in which function they are involved in. So in this case, major facilitator, superfamily, and snare associated, some description is given. Then count in the network, strength, and false discovery rate. So as we know that this particular analysis, how it is done, that you have some background information with you. So in this case also, we have the background information. We know all the proteins which are involved in this particular cluster. And in this case, they are telling you that out of 27 proteins which are involved in forming this particular cluster, 4 are present in my data set or 4 are present in my network. So that is told here. And then it is telling the strength, that what is the strength of these proteins in that particular cluster. So strength will be calculated by looking for the clustering coefficient of each and every node, uh, which means how many connections are formed between these proteins. So if more connections are formed, that means it will have higher strength. If weak number of nodes are present, so it will means its strength will be less. Then false discovery rate. So this is also another parameter to check that whether these are random connections or these are not randomly observed. So that is one measure which tells you so its value should be low so that you can significantly or you can confidently say that these proteins are interacting. Then reactome pathways. So in this case, it will also tell you the pathways in which your query protein is involved along with its interaction partners. So similarly here also in the background list, there are 80 and 110 proteins which are involved in these pathways. And in our data set, we only have 2 proteins. And their strength is also written here, and similarly false discovery rate. Now in this case, statistical background. So in this background, they have selected whole genome themselves. So if you want to change the background, that also you can do. And similarly, you can export the results. So either you can import means export it in a local network cluster, or you can means export their pathways or all the enrichment terms and selected terms only. So in that way, after analysis, you can also download the analysis results. Okay, so now we have till now we have seen the legend and analysis. And in export also, we know that if you want to export the results in the form of table, so what will be present in the table? 2 nodes or 2 proteins which are interacting, they will be present in the table. So that also you can export. Now you can also look for the clusters. So you can cluster the all the proteins which are involved. So in that case, you can go to clusters. It will ask you the options that how do you want to cluster the proteins in the network. So first thing is no clustering. That means the network as shown in the network, that is it. You do not want any other clustering to be used. Or you can select k-mean clustering, which will find a defined number of clusters based on the centroids. So centroids will be like from each proteins, there will be a circle drawn, and From each protein, that circle will be drawn, and if two proteins are, means if their circles are overlapping, then they will be considered to be involved in a single, they will be involved in a single cluster, or you can say they will be categorized together into one cluster. So that is known as centroid. And in this case, we have selected number of clusters to be 3. And edge between the clusters is a, means representation is a dotted line. There are other clustering approaches which are also present. So this is just an example, because in some cases we have multiple nodes which are there. So in that case, these clustering approaches might help you to look for the clusters which are formed between that huge big network. So now it has returned that there are 3 clusters. So out of these 4 genes, means 4 genes, or you can say 4 proteins, they are forming 3 different clusters. In one cluster, there are 2 genes which are present, and this is the description. And similarly, 2 and 3, they are not present in cluster 1. That's why they are lying as separate, they are forming separate clusters. So we cannot actually call them as a cluster, but we can say they are acting as a separate entities, which could not be clusters. So just remember one thing, that my data set has only one single query protein which has only 3 binding interfaces. That's why I do not observe more number of clusters. But if you give any protein which is well known, well studied, so in that case, in each cluster, the gene count will increase. So that analysis will be more significant. In this case, if sample size is very small, so you cannot confidently say that these are the clusters. So this is how you can perform clustering using string. So this is all about string, and uh, I don't think this will be asked in the exam, but it will help you if you want to perform any analysis in future. So do you have any doubts regarding to string? Okay, okay, you asked something. So we can't use average, it will decrease the information. Okay, so what I was saying with the average was that whenever we take average, so there are kind of central tendencies which we use in that mean, median, and mode. So mean means average. So mean is quite influenced by the higher values. So for example, if I have values like 1, 2, 10, and 3. So in that case, if I take average, it gets shifted because of the 10. So if a higher value is present, so my average will get shifted due to that. So in case of means networks also, if any of the node has maximum degrees or maximum connections, so in that case my average degree will get shifted due to that particular node which has maximum number of connections which are present. So that I was saying that average, because previously I commented that average degree, if your average degree is high, then you can assume that there is clustering which is present in the network, because then only average degree will be high. If more connections will be present, that means your network is highly connected. But this is also, there is another condition which is also there, that if in your network one node has multiple interaction partners, if it is having higher degree, then average degree value will also rise. So that's why clustering coefficient is calculated separately, so that you can see that what is the tendency of your network to form a cluster. So that I mentioned. And about the first shell, I think you asked about this, right? Second shell of interactors. So what happens is that there are certain interactions which are not direct interactions, which are indirect interactions. So in that case, there are multiple shells which are present. For example, I have an interacting partner. So for example, protein A is interacting with protein B. So that is my first shell of interaction. That is direct interaction. But what if your protein B is interacting with protein C? So in that case, the second shell will be that my protein A is interacting with protein C. So that is second shell. That means there is no direct interaction, but there is indirect interaction. And why we are considering that? Because in some cases, what can happen is that if you are disturbing one shell, that means if you are disturbing interaction between A, B, then it will eventually also disturb the interaction between B, C. So with that, we can comment that A has disturbed the interaction of B and C. That means A is indirectly influencing protein C as well. So that is known as second shell. So in protein-protein interaction networks, not each and every interaction will be direct interaction. There could be some interactions which will be indirect. So that was mentioned in the shell. So in some cases, you might observe that. So is it clear? Yeah. Now let's move to next question. So the question is, in biological networks, what is a hub? So option A protein with no interactions. Option B, a protein with large number of interactions. Option C, a gene cluster. And option D, a transcription factor. So yeah, the right answer is option B, a protein with large number of interactions, and we have already discussed it. So this is a representation. Now I'll tell you more things about the hub. So I don't think this will be asked, but this is just for understanding. So whenever we are observing a bigger network in which there are small different clusters are present, so in that case, this hub can also have different names. So for example, if my node is present in one cluster which is highly connected, as you can see, there are multiple connections which are present for this particular node, but it is also getting linked to other nodes which are present in different clusters. So with that, we can call it as a connector hub because it is connecting different clusters. So it is having high degree centrality. That means it is having high degree and high participation because it is involved in interaction with multiple different clusters. Now this particular node is having high betweenness centrality. So in the later slides, we will understand that what is betweenness centrality. But now I can simply say that betweenness centrality means a node which is lying in the center. It does not have maximum number of connections, but it is lying in the center such that for each node to reach the another node, that node has to pass through this particular node. So in that case, it is lying in between. So it is facilitating shortest path to be traveled. So that there is one word which is known as traversed. So whenever we study network and we are calculating the shortest paths, so we use word traversed. That means whenever a path is traversed, not traveled. So in this case, whenever one node has to reach to another node with the shortest path, so my means node which is facilitating that shortest path traversed is known as node with the high betweenness centrality. So that is not a hub, but it is having high betweenness centrality. So in that case, if I destroy this interaction, so it will eventually disturb these interactions as well. So that's why betweenness centrality is also another measure other than degree centrality to look for the nodes or to identify the proteins which are more significant. Means by disturbing them, we can destroy the network. Then comes local hub. So local hub means the node which is highly connected, but it is just limited to one particular cluster. It is not connected to different other clusters. So that's why this is known as local hub. So connector hub, local hub, they are two different types of hubs. But overall hub in general means which has maximum number of connections. Now let's move to next question. Okay, what is gene cluster? So gene cluster means these are different clusters now that multiple genes are having means after performing clustering coefficient, these nodes are more likely to form a single cluster. So there are different approaches to find these clusters. So in this case, this is a gene in biological network, these are my gene IDs and how they're interacting with one another. So these are categorized into one single cluster by performing different clustering approaches. So this is referred to as gene cluster, in which there are multiple genes and which are themselves, which are self-contained, which are having multiple connection to one another and not to other genes. So in that case, this cluster is generated. So in journal, if we say hub, so in that case, hub will be like number of nodes, which means all those nodes which have maximum number of connections, they will be termed as hub. But in this scenario, we have clustered this particular network into different clusters. So in that case, we are calling this particular node as a local hub. That means it is just restricted to one single cluster. It is not interacting with other clusters. So that is local cluster. So in a bigger network, there could be multiple clusters. And if it is present only in a single cluster, so this is known as local hub, which is restricted to one single cluster. Yeah, so overall there are four clusters. So it's not forming one cluster. That's why local cluster. So next question is, which tool is widely used for visualizing biological networks? So option A cytoscape, option B blast, option C PFAM, and option D UCSC genome browser. Yeah. So I think this was already covered in the session. So cytoscape, now let's quickly look into the cytoscape. So this is the home page of the cytoscape. You can either download it on your device or you can simply use its online version. So I guess its online version was not available, that's why I did not show it. So you can simply download it. It take a huge space, that's why I did not download it on my device. But if you download it, then what you have to do is there are multiple options which are present with which you can import your network. So how to import the network is that you should have a list of proteins. So in that case, you should have a list of proteins which are more likely to interact with one another. You will simply provide that list of those means two nodes which are interacting with one another to cytoscape, and then it will construct a network for you. So these are different network representations. So this particular A figure, it represents the network which is generated by cytoscape. So in this case, they have used a force directed layout algorithm. So in cytoscape, there are multiple options to change the layout of the networks. So you can also change the layout on means as you want to see the network. So it will change the means how the network appears. So there are different formats with which you can change the networks. So in this case, they have used force directed. In other cases, you can also change it. Then this B figure represents the network which is generated by Arena 3D. So other than Cytoscape, the tools which can help you to visualize the network is Arena 3D, Graphia, and String. So these are other tools which provide the interaction visualization information. And then this particular is the hive plot view. This is also the network representation. And then this particular is the network which is generated by the Graphia application. And this is in the string. So string we have already seen. And in this case, you can also see that there are certain nodes. I think in all the nodes, except these two nodes, there are some structures which are present, which means for them PDB or the structure information is available. So this is the string visualization. So in string, these means visualizations are not that visually attractive. But in Cytoscape, as you can explore different visualization patterns, so that's why people opt for Cytoscape for visualizing the network. So what they do is that from string, they simply download the interactions, protein-protein interactions, and then they upload those files into the Cytoscape and view the network. So in Cytoscape, it also provides you the tools to look for the centrality measures. So in the later slides, we'll see what are centrality measures. But with that, you can simply analyze, you can perform analysis on the protein-protein interactions. So that's why Cytoscape is mostly observed or is mostly used instead of string. Then this F represents the network visualized with the use of arcs. So these are just the representations. This is known as Sarcos. This also you can use in the Cytoscape as well to means view your network in this form. And then this is the visualization of a bipartite graph. So there is one graph in which there are two different sets of proteins which are interacting with one another. So these proteins which are present in this position or in this site, they are not interacting with one another. So this is one of the property of bipartite network, that they do not interact with in one layer. They do not interact with one another, but they interact with the proteins which are present in the another layer. So this is referred to as bipartite network, and this is the representation. So this representation is also seen in Cytoscape as well. So you can use Cytoscape to view the network in this form. Yeah, so now let's move to next question. So what is the clustering coefficient in a network used to measure? Option A, how genes are expressed. Option B, how interconnected a node's neighbors are. Option C, the distance between two nodes. And option D, the weight of each edge. So what do you think we measure with? Yeah, because in certain cases, what will happen is that not direct options will be present with which you can directly understand, but there are some indirectly, sentences are framed indirectly. So you have to means either eliminate other options or you have to see that what could be the possible answer. So yeah, you are right. Option B is right, that it tells you, it measures that how interconnected nodes neighbors are. So if nodes neighbors are highly interconnected, that means clustering coefficient will be higher. That means in my network, these particular proteins are more likely to cluster together. So that is one of the purpose why we use clustering coefficient. Yeah, now there was one paper which was published in the Frontiers. So this is a guide to conquer the biological network era using graph theory, in which they have mentioned that how graph theory helps you to understand biological networks. So in this case, they have completely explained that why should we go for network visualization and why should we use graph theory for studying the biological networks. So in this, they have explained that how that graph theory can help you to study the biological networks in a better way. So first thing, that graph theory in biology. So it is essential for studying biological networks such as DNA, RNA, proteins, and metabolites. So DNA simply here means genes, RNA means different transcripts, proteins, and metabolites. So means we can visually see it and we can study them by using graph theory. And then there are different types of biological networks for which we can apply the graph theory, such as molecular interactions, signaling pathways, metabolic networks, emphasizing their structure and function, and network properties. So, different properties of networks are clustering, motif detection, network alignment, which are crucial for understanding biological processes. So, clustering, as we have already talked, what clustering means. So, by simply calculating the degrees and clustering coefficient, we can see that which of the proteins are more likely to cluster together. Then, motif detection. So, motif are the patterns, which are the short patterns, but they are repetitive patterns. So, that is referred to as motif. So, in that, in networks, we can also detect the motifs. And then network alignment. So if there are two different networks, so we can simply align those networks and we can check that means how two different networks are similar to one another or how they are different from one another. So all these things can help us. For example, if normal state network and the disease state network are there, so that in that case also you can do their alignment. You can check that which proteins have lost interactions. So all those things are facilitated by using graph theory in the biological networks. And they have also discussed about the functional annotation tools such as Panther, David, GSEA for gene list interpretation and biological significance. So these are just the takeaways of this. So what are majorly discussed in this paper? So these are just the points. So I'm not saying that the graph theory is helping you to uh, means perform these annotations, but in this paper they have talked about different annotations by Panther, David, and GSEA, which can also add on to the analysis. Then visualization techniques. So one of the advantage of using biological network is that it helps you to observe visually or observe things visually, which will be easier for interpretation. And then accessibility. So graph theory and network analysis, they are approachable, they're easy to access, and then they are easy to export as well. And from beginner to experts, anyone can use these networks, means observe these networks. So anybody, if you have a file, then you do not have to be the computer scientist or means pioneer of this particular area. You can simply, if you have the idea, then anyone, any beginner can also go for visualization of the network. So that is the accessibility of these networks. Yeah, and so now these are the summary of the paper, that what they have shown here, that how graph theory is helping for the interpretation of biological networks. So one thing is representation, that it precisely constructs a network with nodes and edges, which tells the interaction. So this is about the visualization. Then comes network properties. So graph theory helps you to observe the different properties of that network, such as what is the connectivity. So these are the properties of network, and this comes under graph theory. So connectivity, what is the clustering coefficient or different centrality measures? So as we have seen degree centrality, and we have also checked that there is one another centrality which is betweenness centrality. So in addition to them, there are other centrality measures which are also present. So these measures tells you great detail about a particular node. So in the network, these centrality measures are helpful. And maybe in the later slides, we'll see that how what each and every centrality measure means and how they can help you for the analysis. So with this, they are saying that this graph theory, by using all these properties, you can analyze a biological network in a better way. So that can provide you insights into the functional organization of biological systems and how different components are interacting. Then clustering and motif detection. So that motif and clustering also we can do by using graph theory. So these centrality measures and clustering coefficient, all these properties are generally used to check that where the clustering is happening and what are the motifs which are present. Similarly, network alignment. So you can perform the network alignment. You can also explore evolutionary relationships and functional similarities between two networks. So for example, if I am taking two different organisms, such as human and mouse, and I have one protein, I'm looking for binding partners. So I can also compare that during evolution, whether there are some interactions which are gained. For example, in human, one protein has increased its binding partners or interacting partners, which are not present in the mouse. So with that also we can have some means we can propose some hypothesis or we can comment on something. So that is one of the advantage of graph theory, that you can also perform evolutionary relationship analysis. Then predictive modeling. So topology of biological networks can also help you to predict the effect of perturbations. So for example, how gene knockouts or drugs have influenced the interaction. So with that, you can also do predictive modeling. So if means you are designing some drug and you want to predict the efficacy of that drug. So if you have a hypothesis that this particular drug might disturb the interactions, so you can, by observing the interaction biological networks, you can simply do predictive modeling for that particular drug, that how it is disturbing the network. What are the perturbations? So perturbations are nothing but disturbances in the network, which might lead to lose or loss of interactions or gain of interactions. So perturbation simply means disturbances. Then scalability. So you can also scale because you can also add as many as proteins in the nodes to observe. So it is scalable, that you can increase the data set size by using the modern computational techniques. So that's why I could not download Cytoscape, because although it is scalable, but it requires a huge space. So sometimes it takes longer time to generate a network if your data set is large. So if you have efficient or proper computational measures or equipments that are present, so in that case you can simply means use means simply do scalability as well. Yeah, so this is all about it. Okay. Yeah, so what is the weight of the edge means? Option D in the question. Yeah. So I will talk about that also. So whenever we construct the networks, let's take some example from the network. Okay, in this case, okay, so I do not have any such network where they have mentioned weight. So what weight mean is that there are some interactions which are more influential, or you can say which have more influence. So in that case, there are some weights which are associated with those edges. So for example, if this particular node is interacting with this node, so in this case, they are not providing any weights. I'm just taking an example. So what can happen is that my protein A, when interacting with protein B, so it has a severe impact. Or for example, if it is predicted with more confidence, so in that case, means weight can be given on different ways. Means weight is like priority. So it depends on you that what you are taking, which measure you are taking to give the priority to connection. So for example, if I am giving priority on the basis of connections of this particular node, so in that case, this weight will be like that. So weight is like a priority, which means that how strong is interaction, how strong the interaction is. So for example, if I am giving my weight on the basis of stable and transient interactions, so in that case, if the interactions are stable, I'll give them more weight, which means that I believe that these interactions will stay for longer period of time and these are not just transient. So in that case, they will have more weight. So as in the previous few slides, we were talking about the uh, p-value, that there are some interactions which are simply predicted. They are not experimentally proven. So in that case also, if I am giving weight on the basis of that, so in that case, the nodes which are experimentally proven, they will have more weight. So weight is like preference, or you can say priority, which is given to the connection between two nodes, which tells you that how strong that interaction is. That is simply weight means. So is it clear? Maybe in the later slides, I might have some weight like that. Yeah. So now the next question is degree centrality in a network measures. Option A, total number of pathways in the network. Option B, the number of connections a node has. Option C, the expression level of a gene. And option D, the distance between two proteins. Yeah, this is quite straightforward. So degree centrality measures that how many connections are present or how many connections are there for each node. Now I want to add few more terms. So this is just for understanding, for detailed understanding. This might not be there in your syllabus. So in degree means, so there are some networks, I hope you are aware that there are some networks which are directional. So in that case, when there is some directionality which is present, for example, in regulatory networks or in any network where you have some signal or some interaction which is moving from one side to another side and it is irreversible. So in that case, those networks will be directional networks which have some directionality. So in that case, there are two terms for degree which comes in. One is in degree and another is out degree. So whenever I'm considering my central node to be the main node, so for that we will check that how many of the edges which are going out or which are interacting with the other nodes, and those nodes are not means interacting backward to this node. So in that case, they are referred to as out degree, which are also known as cause. So what my query protein is causing, how it is influencing other nodes or other proteins. So that's why that is referred to as out degree, that is moving out. And then in degree means what is the effect. So what is the effect of other nodes on my given node? So that's why that is referred to as in degree. That means how many connections or how many edges are pointing towards my query node. So that will be in degree. So all the edges coming towards my node is in degree and going out of my node is out degree. And then there is betweenness, which is known as mediator. That means if my node is acting as a mediator between two different clusters, or we can say two different nodes which are lying far apart. So in that case, my node is acting as a mediator, as a central point. So why betweenness is important? For example, if I have a bigger network in which multiple clusters are connected by one single node. So if I break this node or if I knock out this gene, so I can simply break the interaction between these two clusters. So that's why by studying betweenness centrality, we can look for such proteins which can have a drastic or bigger effect on the network. So in the later slide, we will also see another term which is known as robustness. So robustness means what is the ability or means how efficient my network is to deal with the perturbations. So whenever I break or whenever I destroy some interaction, so what is the impact on my network? Whether my network is collapsing or whether my network is still efficiently working fine. So that is known as robustness. So if it is working fine, that means it is highly robust. It can deal with all the missing interactions or with the additional interactions. So for looking for those, for checking whether my network is robust or not, we look for the property of each and every node. So in this case, this node is having betweenness, is having high betweenness. That means if I disturb this node, I knock out this node, so entire network will collapse. So in that case, this node can have influence on the robustness on the of the network. So later on we'll see that means what are the measures, so that in that slide it will be clear. Yeah, okay. So there was another, means I have taken this just to show you that how the average normalized degree is calculated. So this was taken from one of the book, and there's other publication also. So in which they have mentioned degree centrality and its computation for homogeneous multi-layer networks. So there are means this is a multi-layer network. Now in this, these are the name given to each node and edges are present. So in GX, for each node, they have mentioned the degree, and this is the normalized degree, which is means after normalizing, the degree has been calculated. So means for the entire network, they might have performed some normalization to calculate the normalized degree, and ultimately they have performed average of this normalized degree. Now for layer Y, similar proteins are present, but they are lying in layer Y. So in that also they have multiple connections for each of these nodes. They have calculated their degrees and normalized degrees and taken the average degree. And now they have told that over aggregated graph. That means over aggregated graph means, first of all, aggregated means when you are combining two different layers or two different networks. So that is known as aggregated. And why they have taken over means that, for example, I have A which is making connection with B, D and F in this case, and in my GX it is only making connection with B and F. So in OR graph, I will take all the connections. So in this case, it has taken all the connections. After combining these two networks, how many connection A can make is, so all these three connections it can make from GY, and in GX it is making B and F. So in this case, it is acting like a OR gate. So I hope you are aware about AND, OR and NOT gate. So this is kind of OR. But what would happen if you take AND aggregated graph? So in that case, I will only retain B and F. That means, and make sure that I only take those connections which are present in both of them. So in that case, aggregation will only retain B, AB and AF connection. But I have taken OR aggregated graph, so it has also retained the central connection between A and D as well. Yeah, so actually, I have not taken it to tell you about the aggregated graph. I have just taken it to make you understand that how degrees are calculated. So in this case, A will have degree of 2 because it is making two connections. Similarly, E will have degree of 3 because it is making three connections. So this is the way how the degree are calculated, and then their average, how the average is taken. This is just one of the measure. Yeah, now we'll discuss about graph centrality measures. So till now, is there any question? Okay. So now let's just see. So there are other measures also in this. I have listed few. So first of all, first graph centrality measure is node degree. So node degree tells you what is the connectivity of a node. It will be a local measure. Local measure means which is just restricted to that particular node. When we say average degree, so it is a global property because it is taken for entire network, a single value for entire network, whereas local measure means which is just limited to one node. So when we say node degree, so it is a local measure which only gives information of a single node, not of the entire network. Then comes eigenvector centrality. This is another centrality measure. What it takes is that it checks that how many connections my neighbors make. So with that, some score is given to me. So for example, if this node has degree of 4, but in the eigenvector centrality or in the eigencentrality, I will not give the value of 4 to this node. In eigenvector centrality, what I will do, I will check for its neighbors. So there are 1, 2, 3 and 4 neighbors. I will check their connections. So this particular node has degree of 1, this node has degree of 2, this node has also degree of 2, and this node has degree of 4. So I will sum these degrees and divide them by their numbers, means number of neighbors. That will be the score which will be given to this particular node. So that is referred to as eigenvector centrality. And this will be a global measure because means this will be calculated globally also. In this case, I've taken for one single node. Similarly, for all the nodes, this particular score will be calculated, and then eigenvector centrality of a network will be given. So it can be fooled by the fake hub connectivity. So this is one of the problem that uh, means if one of the neighbor has maximum number of connections, so in that case, my query node will also get higher eigenvector centrality value. So in that case, it appears that this node is very significant, is very important in the network. So that is one of the problem with eigenvector centrality. Similarly, there is another centrality which is page rank centrality, which comes under eigenvector centrality only, but it was defined by Google. So why it was given the name as page rank? Because they were looking for the web pages which are more significant or which has the higher value. So in that case, what they were looking, that if one of my page is linked to multiple or is connected to multiple pages, that means this particular page is significant. Because if somebody is searching for this page again and again, that means it has some value which is quite significant. So in that way, they are ranking the pages or web pages. So that's why the name page rank is given. And in this case also, it is also fooled by the fake hub connectivity. For example, if one of the page to which this particular page is connected, if that page is connected to multiple several pages, so this particular page which I am considering will also get higher preference or higher rank due to my neighbor's connectivity. So that's why that comes under page rank, in which the ranking of web pages are done depending on the its importance and number of links which are pointing to that particular page. Then another centrality measure is closeness centrality measure. That means for each node, I will check that how close it is to all other nodes. So in that case, all the shortest paths to each and every node is calculated, and then the closeness centrality is taken. So for this node, how many means how many paths it take, how many shortest path it takes. So for this node to reach here, only one path is there. Then from this node to this, one path. From this node to this, also one path. Means only it has to traverse one single edge. Similarly, to reach this, one. And then to reach this, two. To reach this particular node from here, two paths or two edges have to be traversed. Similarly, to reach this, two edges. So I will add all those shortest paths, and similarly I'll do it for all the nodes. So it will tell me what is the closeness. So after taking their average, so for each node, I can calculate its closeness centrality also, that how close it is to all other nodes and why it is important. So for example, I get some protein which is close to other proteins. So in that case, we can comment that this particular protein is significant in the network because it is closely present to all other nodes. So it is context dependent. Actually, all this analysis is context dependent. It depends that what you are looking for. So in that case, each and every measure is used, means how you select which measure should be selected. So it depends on you that what your analysis is. And at last, there is betweenness centrality. So we have already seen that betweenness centrality means we will check for each and every node from moving from one node to another node, how many times my given node is coming in between. So that will be the betweenness centrality of that particular node. So from this, for this node, to reach these three nodes, it has to travel from this node. It cannot directly go to this node. So that means in this case, the betweenness centrality of this node will be higher. Similarly, for this node, to reach all these three nodes, it also has to pass through this darker blue node. So in that case, this will also have a higher betweenness centrality. But if I consider this node, so it will have betweenness centrality of 0, because there is not any of the node which has to pass through this node to reach any other node. So in this way, betweenness centrality is also calculated for each and every single node, and it will also tell you that what is the importance of a particular node in the network. So these are graph centrality measures. This is just for understanding. You do not have to go into their deep. Okay, so you have asked about the eigenvector centrality. Then the score will be 1 for each if number of edges is equals to number of neighbors. Right. So actually number of edges is not equals to number of neighbors. You number of actually uh, edges are a different thing and eigenvector centrality is different thing. So in eigenvector centrality, you are looking for the connectivity of your other neighbors. So in this case, if I have to calculate eigenvector centrality of this node, so what I will do, first of all, I have four different neighbors for this node. I will check their degrees. So this has degree 1, so I will add 1. This has degree of 2, so 1 plus 2, 3. This has degree of 2, so 5. This has degree of 4, so 5 plus 4, 9. And how many neighbors are there? 4 neighbors, so 9 by 4. So that will come around 2.25, I guess. So that will be the eigenvector centrality value for this node. Yeah, I can add formula for each and every graph centrality measure. If you are interested, I will add it. I thought it will complicate, and means I was thinking that whether I should involve these graph centrality measures or not. Yeah, I will add formula for each and every of centrality measures. Maybe some new centrality measures will also be observed. So yeah, so these are just for understanding or analyzing the network because they provide a detail about the network. Now let's move to next question. I think today I'm moving very slow. So which is the key feature of scale-free networks in biology? Option A, random connectivity between nodes. Option B, few highly connected hubs and many low-degree nodes. Option C, all nodes have equal connectivity. And option D, no edges exist between the nodes. So this we already have discussed. You answered option A. You have to tell in the terms of scale-free network. So when we say scale-free, so in scale-free there are no random connectivity, but in this scale-free there are few nodes which are highly connected and few nodes which are least connected, or we can call them as a low-degree nodes. So this is one of the property of scale-free networks. Means this is the key property actually. This is the term whenever we say scale-free. So we mean this by the scale-free term, and we have also seen that there is power law distribution of the scale-free network. So although random networks means first model for the network was random network model. Dose and Rennie were the two scientists who said that there is a random network, that nodes are randomly connected, but later on small word network model came up, and then later on scale free network model came up. So this is the most advanced model in which there is preferential attachment and a few nodes are highly connected whereas other nodes are low connected, and these are the graphs which tells you about the number of nodes and number of links. So this shows you that maximum number of nodes have lesser links. As we move further, the number of links as the number of links increases, the number of nodes tends to decrease. Whereas in random, it should be normally distributed among all the nodes if the distribution is random. So this is the difference between the random network and scale-free network. Now okay, the heterogeneous connection. Okay. Yeah, so in the random, whenever we say random, so it means that there will be no any bias, or we can say there will be no any outliers. Each and every node has the similar means probability to get connected to other nodes. So that we mean by random in this case. Yeah. Now let's look to next question. So betweenness centrality measures the number of shortest paths passing through a node, the number of direct interactions of a node, the total distance between two nodes, and none of the above. So we have also discussed this as well. Yeah, you are right. So number of shortest paths passing through a node. Yeah, you both are right. Yeah, in this case, I've calculated betweenness centrality. So means what you have to do is you have to check. So I will not individually take each and every node, but I will simply take one of the example. So now let's see for this. So why betweenness centrality is zero? Because there is not any of the node which has to pass through my node 9 to reach any other node. Whereas, let's take the shorter example. So let's take example of node 1. Now its betweenness centrality is 3. Why it's 3? Because means who have to pass through this node to reach other node. For example, if my 5 has to reach to 2, so it can also reach by 3 and it can also reach by going through 1. So one thing is that um, 4 can pass through 3 also and through 1 also. So 1 is 4. Then for 5, I don't know how they have calculated this, because in this sense, 3 is also kind of complicating this analysis. So for 4, if yeah, okay. So you're talking about the between a centrality of sixth node or first node. Okay, you're talking about 5. So actually, I was talking about the first node. Yeah. So for 5, 8 can also reach 4. Yeah, means this is kind of you have to sit for a longer time to analyze that which nodes have to strictly pass through this node to reach the other nodes. So in some cases, what can happen is that there is other node also. For example, if 8 has to reach to 4, then it can either directly pass through 5 or it can also pass through 6 as well. So in this case, 5 is coming in between 8 to 4, 7 to 4, 2, and then 9 to 4 also it can come, 6 to 4 also it can come, and then 3 to 8 also it will come, 1 to 8 also it will come, 2 to 8 also it will come. I think in this way they have calculated this betweenness centrality. So I will provide you with the formulas. Yeah, so this kind of made it complicated because, uh, for example, if 4 has to reach to 8, so simply by passing through 6 also it will take the two edges only, and from 5 also it will take only two edges. So in this case, means it is, I think it will be counted for both for 5 and for 6 also it will add up. So that I was confused with. So you were right. So for 5, 8 to 4, and in this way you have to calculate for each and every node that from moving from one node to other node, how many times this node is coming in between if you are taking the shortest path. Okay, now let's look at the next question. So next question is, what is a common application of gene co-expression networks? So option A is studying protein DNA interactions. Option B, inferring functional relationships between genes. Option C, analyzing metabolic pathways. And option D, identifying mutational hotspots. So you have to tell what is the application of gene co-expression networks. I think it's clear from its name only, because in gene co-expression it will constitute of no, means nodes will only be genes. Okay, so you both have mentioned option A. So in option A, it was protein DNA interactions. When we say gene co-expression, so with which we mean that genes which are co-expressing at the same time. So in that way, we are talking about at the level of genes only. So in networks, we are only taking information of genes. So it tells us functional relationship between two genes, that whether two genes are connected, whether two genes are influencing one another's expression, whether they're influencing the functional interactions between among both of them. So option B is right and not the option A, that is studying protein DNA interactions. So because in this nodes are only genes, so it does not give any information about the protein. Then metabolic pathways, so it is not considering metabolic pathways into the picture, and nor does it tells about mutational hotspots because it is about co-expression. Yeah, there was one paper in which they have very means beautifully explained a human gene co-expression network. So, which was the human gene co-expression landscape confident network derived from tissue transcriptomic profiles. So, means from the tissue transcriptomes, they have taken this information and they've generated the co-expression network. So, generally, it appears like this, in which there are multiple small clusters, which you can see here, and they have divided them into, means they have also done their annotations, or you can say by using the pathway, means because you can also incorporate other databases such as Reactome to annotate each and every protein which are present in the cluster. So in this, they have seen that these cluster 2 is involved in immune response. So means how do they identify this? Firstly, what they do is they are simply firstly clustering all the proteins which are present in the network. Second thing, what they are doing is that for each of the cluster, they are checking that how many proteins are there and what are their functions. So after getting their functional means functions, they are comparing with background. So in this case, for example, all these proteins are involved in the immune response. So overall, if my 60 proteins are involved in immune response, and in this cluster, if I observe 20 proteins, so I will see that what is the means either it is confident that they are involved in immune response. So what is the enrichment of these this proteins in the cluster to fall in the immune response? Because it is not always the case that all the proteins will only be associated in immune response. There are cases where one protein can have multiple functions, or there are cases where they may not be involved in immune response. But if majority of proteins are involved in immune response, so this cluster will get annotated as immune response because we have to select one of the pathway or one of the term for a cluster. That's why they have taken these terms. So in this way, this is how the gene co-expression network appears, and this is telling you about the functional interactions, or you can say this is providing you the functional information for the genes, that what are the function these genes are performing in each cluster, which are expressing at the same time. Okay, so let's look into next question. Next question is, which molecule is typically responsible for signal transduction in cells? Option A lipids, option B proteins, option C sugars, and option D water. Yeah. So the right answer is option B, that is proteins. Now next question is, protein kinases are enzymes that regulate the biological activity of proteins by phosphorylation of specific amino acids. Which among the following is not true regarding the protein kinase activity? So now you have to tell that which is not true. Option A, mutation in kinase domain can create a new phosphosite for protein kinase. Option B, mutation in the kinase domain can create a new kinase binding motif. Option C, mutation in the kinase domain can lead to the change in a kinase binding motif. And option D, mutation in the kinase domain does not affect the activity of kinases. Yeah, you are right. So option D is the case. So mutation in kinase domain does not affect the activity of kinases. It's false because mutations, they can either lead to change in the binding motifs, or they can also create new motifs because they are changing the sequence levels. So they might change the binding motif in which maybe some new partner can bind to them, or maybe they can influence the we can say the strength of interactions which is there, or means strength of means this phosphorylation they can change, and they can also create new phosphosites. For example, if there are certain amino acids which undergo phosphorylation, so if one particular amino acid is changing to the amino acid which undergoes phosphorylation, so in that case they are creating new phosphosites. So option D was false. Now question number 13, which of the following statements are true regarding the single point mutations in the genome? Option A, it can change the amino acid sequence of a protein. Option B, it can result in a synonymous mutations if located in a protein coding region. Option C, it can eliminate a site for post-translational modifications. And option D, it always leads to a change in protein function. Yeah, so you're right. All ABC are true. And it's quite simple. So no need of explanation is there. Because why option D is not right? Because they mentioned it always leads to change in protein function. So means it's not always true. It can lead to change in function, but not always. Now question number 14. Which of the following features are affected by MIMP, which is mutation impact on motif phosphorylation tool? So you have to tell what are the features which this offers. Option A, it can assess the impact of mutations on kinase binding motifs. It provides predictions about potential protein-protein interactions. Option C, it can identify mutations that may lead to altered phosphorylation patterns. And option D, it offers structural modeling of protein complexes. Yeah, so you're right about the option A, C, and B is like it provides the prediction about potential protein-protein interactions. So means it does not exactly tell about you of the protein-protein interactions, because it tells about the uh, binding post-translational modification. Actually, it incorporates the post-translational modification information. So it uh, does not provide predictions about the potential protein-protein interactions. That's why this option is not right. And these are the answers because this question was there in your previous year assignment in which they have mentioned option A and C to be true. And then option D is completely wrong. There is no question for this. So in this case, I have taken the paper which was published in 2015 in which they have explained MIMP, which predicts the impact of mutations on kinase substrate phosphorylation. So as we all know that protein phosphorylation is very important for different pathways, And if we talk about disease, so these phosphorylations, if they are means if they are impaired or if they are disturbed, then they lead to disease. So in that case, they have developed this MIM to look for those sites. So this is a machine learning method in which they have used machine learning algorithms to predict the impact of missense single nucleotide variants on kinase substrate interactions. So how the kinase and substrate interactions get changed with the missense single nucleotide variants, which are SNVs, and missense means which are leading to change in amino acid. So they're not considering synonymous mutations where there is no change in amino acid. So with that, they're predicting its impact with the kinase. So okay, I thought you asked some question. Yeah, so MIMP analyze kinase sequence specificities and it predicts that whether SNVs will disrupt the existing phosphorylation sites or whether it will create new phosphorylation sites. So in that way, it discover mutations which can modify the protein function by altering the kinase networks. So kinase network means when kinase are interacting with the protein, so how those interactions are getting changed. So that means altering the network of kinase, and it provides insights into the disease biology and therapeutic developments. So means how different therapies can be developed by talking about the phosphorylation sites. So in this case, MIMP is dedicated to protein phosphorylation changes with respect to the missense single nucleotide variants. And there is a workflow of MIM which also I have listed. So what it does is it begins with constructing models for known kinase binding sites. So in this case, the serine and threonine, they are more likely to get phosphorylated. So firstly, they look for those sites which are known to be kinase binding sites. Then they predict the effect of cancer mutations on phosphorylation sites. So in this case, they have constructed a model and now they are identifying. So they have constructed a model and then they look for the predicted so density and peptide score. So means they have performed some analysis to check that how the change in this particular amino acid at this site is influencing with the binding of the kinases to this particular protein. So with that, they have taken some scores. Uh, then they have performed these mutations. So means this score has also been previously taken for the negative and positive. So this was already information which was already available. Then they have performed mutations at different sites where they have altered some of the amino acids. They have performed missense single nucleotide variations. So these are wild type phosphosites, and after performing these mutations, these are mutant phosphosites. Now they have identified PSNVs and they have predicted the rewiring events. So, means after changing the amino acids present at those phosphosites, what can happen is that now my binding of kinase might change. So in this case, they are just influencing the neighborhood, means the neighboring amino acid positions, I guess. They're not influencing serine and threonine, but they are disturbing their neighborhood sequence. So what we know that kinase bind to protein, and in the previous PPIs we have seen that protein-protein interactions, they're physical interactions, and they are quite specific. So with specific we means that there is a site, there is a peptide sequence, or we can also call it as a motif which is responsible for binding. That is not just one residue which is responsible. We know that serine threonine might get phosphorylated, but they are not just a single amino acid which is responsible for binding of kinases, but there are some neighboring residues also which are involved in the interactions. So in the mutations, what they have done is they have varied those uh, surrounding amino acids so that the binding interface or the binding peptide sequence get influenced, and then they are comparing that how this binding pattern, how this means interactions get disturbed. So whenever we look into the interactions of the networks or interaction network, so we call them as rewiring. Rewiring means after performing mutations, how my network is changing its interactions. So that is rewiring. Now interactions are changing, they are rewiring themselves. So how many potential losses are happening and how many gains are happening. So gain means gain in number of interactions, which simply means gain in the degrees. So that thing is taken into consideration and then conditional probability has applied. So these are some statistical measures. You do not have to go into the detail. But overall, what they are telling you that how the mutations are influencing the interactions. So with which they have performed their interpretation. And in option, so in the B, they have shown the analysis of mutations from TCGA, and they reveal categories of phosphorylation gains, losses, and switches. So this is mentioned here that how many times gain has happened, how many times there is switch, and there is loss. So means switch in the other type of kinases which are binding, and then rewiring events are mentioned in this particular C. Yeah, so means I won't go into much detail of these experiments. These are not required, but overall, what I want to tell you from MIMP is that it just looks for the SNVs which are responsible in changing the interactions. And how they are changing interactions, they are focusing on the phosphorylation sites. So they are interested in the kinase binding. That is one thing, and they are predicting the influence of change of those phosphorylation site motif. So I hope it is clear till now. And don't worry about all other details which were mentioned in the paper. They are not required. And if you want to study them, then I have provided the link. You can read their paper. Now let's look at question number 15th. So there are many tools. Okay. So SNV is equals to SNP. So no. Single nucleotide variations and single nucleotide polymorphism, they means often overlooked, means they many people think that they are the same, but single nucleotide variants and single nucleotide polymorphism is a different thing. Single nucleotide polymorphism are the, whenever we say SNPs, we call them as SNP because they are associated, they're not always associated with the disease and they're normally observed. But when we say single nucleotide variations, in most of the scenarios, means these single nucleotide variations are linked with the disease. That this particular variation means SNV are the later term which are given. So means when you say SNP, it is constant among the population, but SNVs are rare. SNVs are not that much prominent in the population, so these are not same. Similar is the case for copy number alteration and copy number variations. So this is simply a term, but they are not the same. Yeah. In most cases, when we say SNV, so we generally label those SNP as the variant which is involved in the disease. So that is one another thing which you can take. So now question number 15. Okay, you already answered option C. Yeah, so there are many tools. You have to match them. Now let's see. I don't want to waste more time. So the right answer is actually option B, in which phospho-SNP is the database for the mutations and mutations predicted to impact phosphorylation and phosphorylation status of proteins. And when we talk about active drivers, so it predicts the active phosphosites in the protein that are significantly mutated in cancer genome. So when we say active driver, so it is specifically talking about the cancer genome. So that's why don't get confused among phospho-SNP and active driver. Phospho-SNP is for the proteins, how the mutations are impacting the phosphorylation status, whereas active driver is specifically for the cancer genome. And then PTM where is meaning that means it is intersecting known synonymous SNPs and PTM sites, that what are the post translation modification sites and how it is linked with the known synonymous SNPs. So in many cases they have taken SNP also. So in many people call them SNV when they talk about diseases, but in this case they have also talked about, they have written as SNP, so don't get confused. Means many, you will see that people are using it interchangeably, so that is not the problem if even if you write SNP. Now fourth option was phosphovariant, so it is a database for definite and possible variants changing the phosphosites. So it tells about the variants, all the possible variants. Okay, now let's look into question number 16. So is it clear? Because I think we'll go through active driver also in the later slides. And yeah. So the question number 16 is, sequence motif analysis is a test for the enrichment of amino acid pattern surrounding the phosphorylation sites by comparing against a background dataset. Which among the following tools can be used to perform sequence motif analysis? Option A, motif X, option B, phosphosite plus, option C, mascot, and option D, sequest. So what do you think would be the tool which could be used for the sequence motif analysis? Option A. So you have to tell answer from 1 to 3. Acha, okay. Yeah, so I think Srijita also wanted to say Roman I as option. So first option is right. That means motif X and phosphocyte plus. And as mascot and sequence we have already seen during mass spectrometry lectures. So these two options are right. In this I have explained that why so what motif X provides. So it is used to perform sequence motif analysis, and phosphocyte plus is also a database which we have seen previously that for the phosphorylation sites. While it doesn't directly perform motif analysis, but it can be used to provide data on phosphorylation sites indirectly supporting motif analysis. So it does not directly provide to perform the motif analysis, but in these options we have selected AB because there is no option which is only mentioning option A. But if this question was given as a MCQ and you have to select among motif X and phosphocyte plus, so motif X will be your answer. Okay, so now in this I have taken the paper which in which they have talked about motif X. So in this they have said the biological sequence motif discovery using motif X. So how motif X identify the motif? So they have mentioned that it is a simple interface to extract statistically significant motifs from large data sets such as mass spectrometry MSMS. So when there are two different mass analyzers are present, post-translational modification data and group of proteins that share a common biological function. So for them, whenever you are using the motif X to identify their motif, so means that they have mentioned, and they produce both syntactic and image-based motif results and statistics. So how do we identify the motifs? So first thing is that if I already have the information which is experimentally present, or another thing is that if we have an image, if we have two structures and we know the interaction positions, then also with the image also we can predict that binding motif. So in this, they have mentioned that there is a foreground data set, and in the right side, it has a background data set. So we have information of MSMS peptides. We have a FASTA sequence, which will be a sequence file. This will be a peptides sequence, and this will be a FASTA sequence, and then there will be a pre-aligned sequence. So in this case, it is elaborated here that user provides a foreground and background data set for analysis and other analysis parameters which are presented in the middle dash box. The use of MSMS dataset always requires the specification of the organisms it comes from. So you have to specify that for which organism you are doing it. Then comes standard database libraries, which it is mentioned here, SDLs, which are for common model organisms, are provided on the server for this and other purposes as shown. Light green boxes show processing steps, while non-colored boxes indicate data inputs, data outputs, and intermediate results. So these boxes are the processing steps. That is pre-alignment. In this case also there is pre-alignment, then fixed width, and then motif X core engine was there, and then it will generate a motif information. And in this case, they have mentioned, so whenever you are applying some conditions such as central residue to be serine and width is 15. So means after having all the sequence level information and performing the alignments, they are looking for the motifs. So how motifs are actually found is that we for a particular to call that this particular motif is involved. For example, if we have a protein, so in that particular protein there are multiple other proteins will also be there which are related to that family of protein. So for all those proteins who are interacting, for example, if I want to look for the kinase binding motif, so how I can say that this is the binding motif? For that I have to see that which other proteins my kinases interact with, and for all those proteins I should have the information of the means their interaction interface. So for all those proteins, I will align those positions, or I align those sequences where the binding is happening. So in this case, MSMS peptide information is taken as we discussed in previous case that it will tell us, for example, in a complex, which are the two proteins which are involved in the interaction. So it is telling me PPIs, where the protein-protein interaction is happening, which are the peptides which are involved. FASTA sequence will tell me the complete entire sequence, FASTA sequence, and then there will be a pre-alignment which is already available of those sequences. So I will compare, I will give score to each position of that particular binding site. So I will see that which amino acids are present or is occurring commonly in all those different sequences. So with that, I will give score to each site. With that, motif is actually identified that this is the binding motif. Because in, for example, if we have multiple different proteins, so binding motif is not always means 100% identical. Sequence can vary to some extent, but although it still maintains that interactions. So this is a way how the motif X identifies the motif sequence. And if you want to go into more detail, then you can go through this paper. Link I have already provided. So this is just to tell you that how motif X discovers the motifs. And in this, they have provided a table. So if means you have different objectives, then what data actually you required in a foreground. So to find statistically overrepresented motifs from row MSMS post-translation and modification sequencing data, your foreground data should be MSMS data. And this is the means example input of the data. So in this case, star and hash, they represents the post-translation modification sites where that is happening. So this is the example data, how the data will be there. These are the peptide sequences and these are the sites where PTMs are happening. So this will be the example. And then next, if your objective is to find statistically overrepresented motifs from previously aligned sequence data, for example, substrate of a kinase where the central residue represents the modified site. So in this case, what can happen is that if you have from different organisms, if you take some protein which interact with kinase, and then you align that position, that where the kinase is binding, you align that sequence, so you will get a pre-aligned sequence. So with that, you will be able to find that where the changes are happening. So for example, in this case, they are saying that central residue is representing the modified site. So most likely what has happened is that in the central position, maybe some amino acid is changing among all of them. Or it can also be other way around, that if some residue is changing in the center, so that might not allow kinase to bind to that particular protein and they disturb the interactions. So for this, you have to provide a pre-aligned data to find such means to identify that what are the sites which are getting changed. So for that, you have to perform the alignment. So that's why the data will be pre-aligned to find statistically overrepresented motifs directly from the whole protein sequences. So for that, you have to provide a FASTA sequence. So this will be an entire sequence of the protein. And then text. So to find statistically overrepresented motifs from a linguistic text file. So when we say motif, in case of protein, it will be a protein sequence, but in this case, they have taken an example that if you have a journal text, so motif, what motif will be? Motif will be some word or some means group of words which are means repeating again and again. So in that case, if there is a text, it will try to find statistically overrepresented, means which word are appearing again and again. So that it will search here. Okay, so this is for the identification. So for statistically overrepresented motifs, you have to provide a text foreground. And then I looked into their server also, that how means currently their server is not working, but when they mentioned about their server in the paper, so this was the homepage of the server, and they mentioned that you have to upload a file for the foreground data set. So in the previous, as we have seen, that if you have those objectives, so what should be your foreground? So similarly, you can upload your foreground in this particular box, or you can either upload your file also. Then you have to select. So in this case, the data was MSMS data, and they have selected that it is IPI human proteome data. Then there are basic options and advanced options. So basic options are already set by default, that what should be the significance. So what will be the results? So it will means give you the results in which firstly it will mention the job name and everything, that what is the data set, what is the width. So whatever you have selected in the basic options, that will be listed. Now later on, how motifs will be identified. So in this case, it might have taken these 1, 2, 3, 4, 5, 6, 7, 8. So 8 sequences might be compared, and then the probability of amino acid at each position is calculated. So in this case, as you can see, there are multiple amino acids which are listed here. So if the size, or you can say if the width of amino acid is high, that means that amino acid is more frequently occurring at that site. So as you can see in this case, S and P are written big. That means in all these protein sequences, S and P were always present at this site. So in this way, motifs are generally when you give the pre-aligned data, or if you have a peptide data from different samples or different PPIs were taken. So in that case, you compare those sequence positions and you check that which amino acids are is constantly present and which are changing. So with that, motif prediction is done. So in this case, for each of these, the motifs will be identified. The score will be given. Foreground matches foreground size, background matches and background size, and fold increase. So each and everything is calculated here. And then in this, they have mentioned in detail about the result, that how many peptides are involved in the interactions, how many peptides are present in the original data set, and how many peptides in the original data set that are unique, how many peptides are found in the database, that is IPA human FASTA, how many peptides not found in the database. So because you already are providing the background information, so they're comparing among foreground and background. So with that, the prediction of motif is happening, and how the change, how it leads to change, so that is being calculated here. Yeah, so this is about the motif X, and I don't think you have to go into the detail of motif X, but this was just to see so that you could remember it. Now, next question is, active driver database is a good resource to study the mutations and post-translational modifications. Which of the following statements are correct regarding the active driver database? So first option is, it predicts the impact of mutations by analyzing gains and losses of kinase bound sequence motifs using MIM. Option B, it contains the information of somatic mutations identified from 9,000 tumor samples. Option C, it is a SNP database from lung, heart, and blood disorder patients. And option D, it contains the information about the drugs that target upstream enzymes of PTMs. So you have to, from these options, you have to tell which one is correct. So according to you, second option. So option D is also actually right. A and B are correct. And option D is also correct, that it contains the information about the drugs which generally target the upstream enzymes of PTMs. And option C is wrong because it is specifically focused on the cancer datasets, so not on the disordered patients. Now, next question is, what is the role of phosphatase in signaling pathways? So, as we know about kinase, so for the phosphatase also you have to tell their role. Option A is to activate kinase. Option B, to dephosphorylate proteins. Option C, to inhibit protein translations. And option D, to degrade proteins. Yeah, you're right. So the right option is option B, to dephosphorylate proteins. They does not activate kinases, but they dephosphorylate the proteins. Phosphatase, the enzyme which break the phosphorus bonds. So it will dephosphorylate the proteins. Yeah, now there are some proteins which are RAS proteins. So they are involved in which type of signaling you have to mention. So option A, apoptosis. Option B, metabolic pathways. Option C, growth factor signaling. And option D, immune response. So actually RAS proteins are involved in the growth factor signaling and not apoptosis. Then question number 20, mutations in which genes are frequently associated with cancer? Option A, BR means BRCA1. Option B, MYC. Option C, KRAS means KRAS. And option D, all of the above. The right option is all of the above. Yeah, this question means if you are more into the literature studies, then you might be aware of MYC and KRAS also, because these KRAS, as it is, means the type of RAS only, comes under the family of RAS. So these are involved in growth factor signaling. So if all these genes there is mutation, so they will affect, because they will impair the apoptosis, they will increase the cell growth. So that's why they are associated with cancer. So that's why option D was correct. No, 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 no. These will not come. This is just for understanding. I don't think they will come. These are just means kept by me. They were not present in previous assignments. Okay, we'll just quickly go through. I think these questions are not that important. I'll provide you the slides so that you can go through these questions. Now, all these questions are not important. Um, yeah, so I will talk about this question, which is important. So, which of the following statements about common post-translational modifications is correct? Option A, phosphorylation typically occurs on serine, threonine, and tyrosine residues. Option B, acetylation commonly modifies lysine residues to regulate protein stability. Option C, methylation primarily affects arginine and lysine residues, influencing protein-protein interactions. And option D, glycosylation is exclusively a modification that occurs on hydrophobic amino acids. So now you have to tell that which among the following statement is correct about the post-translational modifications. So actually option C is also correct in addition to option A and B. I have kept this question because you have already looked into phosphorylation and acetylation as PTMs, and you should be aware that which of the amino acids are more likely or which occurs or which undergoes these PTMs. So serine, threonine and tyrosines, they undergo phosphorylation, lysine undergo acetylation, and arginine and lysine, they also affects the means they also undergo methylation, which will influence the protein-protein interaction. So keep methylation also in mind in addition to phosphorylation and acetylation. Okay, so in this case, they mentioned that it occurs specifically on hydrophobic amino acids, that about glycosylation. So for the glycosylation, it is not actually happening on hydrophobic amino acids, but it also happens on asparagine, serine, threonine residues, and not specifically the hydrophobic ones. So keep this point also in mind. So in PTM, phosphorylation, acetylation, methylation, and glycosylation all comes under post-translational modifications. And I hope now you will be aware of the amino acids also, which can undergo these, means which act as a site for these types of PTMs. So in last, I have kept the reactome server. So this is the homepage of Reactome. Now we will quickly see that what we can do in Reactome. So first option is pathway browser, in which you can means observe or you can view the pathways. So this is the means description of entire. Whenever you click on the pathway browser, so it will take you to this particular screen in which there are different event hierarchies are present, different events are there. So this in this they have talked about all the pathways in the hierarchical pattern. So whenever you will click on any of the pathway, then it will list out the details of that particular pathway. That's why they have called it as hierarchical. That stepwise they will tell each and every step of that pathway, that in cell cycle what are the later stages of that cell cycle. So in that way it is like a tree. So this is the home, and here you can select a species, Homo sapiens or any other species in which you are interested in. This particular is the display pane in which it tells you about different, means from these only they have listed all the pathways. And then if you want to fit it to a page, so these are different options. If you want to open the diagram, these are for the illustration. If you want to export it, this is for the key. So key will tell you that what each and everything means. So whenever you will view their, means this is just a network representation, but there is another representation which is also available. So in that case, to know that, for example, circular node, rectangular node, what they mean. So for that, you can go to key and it will tell you the detail of the picture which you are seeing here. So in diagram search. So in the detail panel, why I have kept this page actually, because in your assignment this was mentioned that in the details panel you will see the expression. So currently their server does not have this option of expression in their server, but previously it was there. So now you will not be able to look into the expressions, but there are alternative ways with which you can find the expression value for each and every gene or each and every protein. So I will tell you later. Yeah, so now what I have done is that um, You can select any of the, so this is the current, how the current server appears, and these are different pathways which are listed. Now what we can do is, if we select the autophagy, then this will be, now here they will focus, they will enlarge this particular window and they will focus on the autophagy. Now in autophagy also there could be multiple options, but before going to that, here you can see it is a visual cartoon diagram of a pathway. So if you click on this, then you can select key and check that what this figure means. So in the description, it will tell you the summary of autophagy, what is autophagy. You can read about it from here. And if you click on this particular image which is appearing, so this will make up a cartoonic figure or cartoonic pathway, that how different steps are involved in the autophagy. So this, you can see these are the proteins which are listed in each step, and what is this means, how the stepwise the reaction is happening. So this is for that purpose. Now in autophagy also, if we click on this plus, then it will appear, it will show multiple other options which are under autophagy, such as macroautophagy, chaperone mediated autophagy, late endosomal microautophagy. So after that, if you select macroautophagy, then there will be multiple reaction steps which will be present. Now in this case, with each of your option, with each time you select any option, it will kind of elaborate your analysis. So in this case, it is elaborating my pathway step. Previously, as you can see, in this case, they are simply listing the nodes. And now if I further click on the macroautophagy and I click on the active mTOR C1 binds, so in this case, this particular pathway will be further, means you can say expanded. Now they are specifically highlighting this particular step. So these are the reaction steps which are involved in macroautophagy, and you can click on any one of them, and then it will tell you that where this network, where these nodes are lying and in which reaction they are involved in. So if you click on that key option which I mentioned earlier, so it will tell you the meaning of each of these boxes and each of these edges, what they mean and what this dot means, what these boxes means. So these are the complexes. So all those things can be seen from the key. Okay, now if you want to search for the proteins, that what are the proteins which are involved in this particular pathway. So what you can do is that previously we were on the description. Now what we have to do is we have to move to molecules. In molecules, you will find chemical compounds as well as proteins which are involved. So there are 18 proteins. From this, you will be able to see the UniProt IDs as well as the gene names. So from here, you can select any gene name. For example, there was a question in the assignment in which they have given some gene names. So if you cannot directly find it, so in that case, what you have to do is you have to go to molecules and then search for that particular, for all those gene, you have to check that which of the gene is present. So with gene, I mean which of the protein is present. Now, for example, if I select my 43504, which was present somewhere here. So if I click on that, if I click, sorry, here only. So if I click on this plus, I think this one is some one other UniProt, not this one. But yeah, if you click on this plus, so it will elaborate it. It will provide each and every detail which is associated with that particular UniProt ID, such as RefSec, PDB. So PDB is about the structural information. All those informations will be given here. And there is other option which is gene cards. So in gene cards, it will further elaborate more detail about that particular protein or that particular gene. So in the later slides, we'll see that also. Now, after molecules, you can also go to structures. If you want to means know the structure of each and every protein which were involved in the means macroautophagy, that particular means autophagy steps. And then you can also perform the analysis. So for analysis, you have to click here. And analysis, you have to choose the data file, or either you can paste the genes for which you want to perform the analysis. So in this case, I have pasted these genes. After pasting, you have to click on continue and then select your preferred options. So in this case, this was by default selected, project to human. So in this case, what it does is that if any of the gene, for example, if you provide a ID, so if any of the gene For example, some kinases has some ID and your ID is related to some other species. So what it will do is it will change that ID to human. So to a human equivalent of that particular gene because you are performing analysis on human. So that's why what it means project to human means that any of the gene ID which is not belonging to human but that gene equivalent is present in human, so it will means convert it to the human equivalent of that particular ID. Now you have to click on the analyze and this will be the result of the analysis. So it is telling me that of that gene list which I have uploaded, these are the pathways in which those genes are involved. Then in entities found means how many of those genes are involved in all these pathways. So this is entities found, and then entities total means in the background how many entities are actually involved in these pathways. Then entities ratio. So out of all the entities, what is the ratio of entities in the platelet B granulations? Similarly, p-values, FDR, and reactions which are found. So how many reactions that particular entity or that gene is involved in? How many total reactions are involved in this particular pathway? Then reactions ratio, species. So this is the result of my analysis. So this is just to check, for example, if I have a list of gene and I want to check that how in which pathway these genes are involved in, so I can perform this analysis and ultimately I can also download my results as well. Okay, so in analysis also, what we have done is analyze the gene list. Another is analyzing gene expression, and in that they have also provided a guided tool. So I think it's already 8:12. So should I continue or do you guys have some other works to do? Okay. Yeah, yeah. Yeah, so analyze gene expression. So in this, everything which will appear is from the guided tools. So you can simply click on this link and it will take you to guided tools. So if you do not have your data set with you, so you can go to guided tool. It will tell you step-wise how we can perform the reactome pathway analysis and analyze the gene expression. So first of all, there are three different select methods in which they have selected camera panel to select the camera analysis method. So for first analysis, people use this analysis method. And they have also mentioned that what this select method does. So it is a gene set analysis algorithm, which is similar to classical GSEA gene set enrichment analysis algorithm as implemented in the Lima package. So these are the methods to identify the gene set enrichment. Now you have to click on continue after selecting that. These are the parameters which you can, if you want to select, you can select, which are the maximum missing values and discrete normalization function, continuous normalization functions. So after selecting, these are by default present, but you can change these parameters if you want. Then you have to click on continue. Now the step 2 is to add and annotate your data set. So now you can add your data. Means if you have the RNA-seq data in which there are row counts or normalized RNA-seq data, or you have a proteomics intensity data coming from IT, RAQ, and TMT, or you could also have a spectral counts or microarray data. So these are the local data which you can upload. In the example dataset, they have provided the exemplary dataset. For example, you do not have your own data, then you can select the example dataset. And then the third was public data, in which publicly available dataset. So species, and then you have to enter the keywords. Then they will tell you the publicly available datasets. So that also you can select. Similarly, expression atlas, single cell expression atlas. So in them, if there is dataset which is available, that comes under the public dataset. And from here you can search for the organism. And this is the example data as I've discussed for the melanoma RNA-seq example and melanoma proteomics example. So this will be from mass spec and this is from the RNA sequencing. Now I have selected the this melanoma RNA-seq example. So if you click on that, now it will load the data set in this form. So this is the data set, protein IDs, conditions, and cell types. Now you simply have to upload this table for your analysis and then you have to click on continue. Now you have to select the comparison factor. So in this case, condition they have selected because we are comparing among the condition of a patient. And the groups, so we have to distinguish among two groups. So first is MOC group and then is the MCM. And in this they have selected the covariates. So you don't have to go into actual detail of this. This won't be asked, but this is the guided tool, and then you have to save the data set. You can also add the another data set if you want to perform the comparative analysis among data sets. Otherwise, you can simply continue it. Now, third step is analysis options. So it will ask you that create reactome visualizations, or you want to create reports, or if you want your analysis result to come into your email ID, so that also you can select here. And then you have to continue with this. Now it will take some time to perform the analysis. And after performing analysis, it will tell you that your analysis has been finished. You can click on the results, and this will be your results in which different pathways will be listed, their entities will be present. Now, what is the difference between the previous one and this one is that you have taken the condition as a parameter. So it will tell you that what is the conditions, means how that particular gene is, either it is involved in the MOC condition or it is leading to MCM. So that thing will be reported here from the melanoma RNA-seq data. But in this case, they are generally representing the pathways in which those genes are involved in. So by clicking on these entities, you can look for the genes in the melanoma which were found to be involved in this pathway. And you can also download the results. Means, for example, if you want to perform the analysis yourself, then you can download the PDF, or these are different formats in which you can download. You can also download the pathway diagrams. Yeah, this is all about the reactome. And one quick thing which I want to add is that in your assignment, you were asked about the expression. So if you do not have the expression information, what you can simply do is you simply in the proteins, you have to firstly look for those proteins, those IDs you have to search for, and then you have to click on plus option, and then it will give you external cross references. So as that TA also has mentioned, that either you can go to UniProt, or another thing which you can do is you can go to gene cards. So if you click on the gene card option for that particular UniProt ID, so it will take you to this home page in which there are multiple options which are present. So you can simply jump to sections. In this case, there is a section named as expression. You can jump to this expression. It will take you to this particular section in which it will tell you all the tissues and what is the expression values. So now from these graph, you won't be able to interpret. So what you have to do is you have to move down, and in which they will tell you mRNA expression in embryonic tissues and stem cells from. So in this, they will mention you the tissues in which the expression is, means in which there is a significant expression. So in this case, that particular UniProt ID is expressed in the brain and testes. So similarly, the gene which was present in your assignment, for that also I have performed this analysis, and these are the results. So skeletal muscles, testes, and heart. So these are the boxes which represent the gene names, and this is telling you about the tissue in which that particular gene is enriched or expressed. Yeah, so this was all. I hope it helps you with the assignment. So this was all about today's session, and sorry for taking longer time, because I was not aware that these questions will take time. Okay, you can ask your questions. So background data is actually the data which is already available in the string. For example, in a human genome, we already know what are the proteins which are interacting with one another. So that act as a background data. For example, if gene A is mentioned, means because I mentioned that how they obtain the interaction information, they take the information from the experimental evidences, as well as they also incorporate the information which is theoretically gathered, such as in the literature. So from all those information, they construct the protein-protein interaction for the entire human proteome. So from there, if I'm giving some gene or I'm talking about some pathway, so it will, what it does is that from that background protein-protein interaction network, it will take a small section of those proteins, that how they're interacting with one another. So for that only they're picking it. So in that case, the background data will be my entire human proteome PPIs. So they are acting as a background data, which string already contains. as BLAST. So in BLAST also, as we know that it already have all those sequence information, and what you do is you give your unknown sequence and you are trying to align it and you are trying to identify that to which organism this sequence might belong to, or to which gene or protein this sequence might belong to. So similarly, you're doing that thing here also, that in string already PPIs are available. You are giving your list of proteins, or you are giving a single protein, or you are giving a pathway, and you are just checking that with this protein, how many proteins are directly interacting, or second shell interactions can also be gained here. So in this case, that is acting as a background data. Okay, so differentially expressed data set is like, it depends that for which data set you are talking about, because differentially expressed means which are variable in their expression. So in which context you are talking about the differential expressed data set? About string or some other? Can we use string using it? So string simply takes the list of the genes or list of the proteins for which you want to predict the protein-protein interactions. So what will be there in the differential expressed data set? Because when we say differentially expressed, what we if we are talking about genes which are differentially expressed, so what we talk is that in a normal state, in a disease state, if I give a list of genes which are differentially expressed, which means that these genes they alters their interaction partners. So that is generally referred to as differentially expressed genes, or similar for the protein as well. So string is simply taking the list of proteins, or it can take a single protein. So in that case, you can use the differentially expressed data set. So that you are asking, or you are asking that can we use the differentially expressed data set as a background? Because what can happen is, one more thing which I want to add is that generally background data is entire human proteome interaction data set, but you can change the background as well. So background means whenever you are giving your list of genes, you are just trying to check that from that background, what is the enrichment for this particular question or this particular query which I have asked. So if you are changing the background, that means you are changing the comparison set. So in that case, differentially expressed data set. So for example, if you only want to limit your analysis to those differentially expressed genes, so in that case, you can select differentially expressed data set as a background. So in that case, it will only compare for those gene sets. So I mean, if you take entire human proteome, so how it will calculate enrichment? What it will do is, for each pathway, it will check how many total genes of the entire human genome is involved in this pathway, and from your data set, how many genes are observed in this pathway. With that, it is calculating enrichment. But if your aim is to study differentially expressed genes only, and you want to check that these genes, either they are present in that, means in differentially expressed, what is the enrichment of those genes in this pathway. So in that case, a background will be differentially expressed data set. So what it will do is the averaging it will doing. So the average which it will do is with that limited data set. So if your aim is to specifically look for a single, means disease x means differential expression, then background will be differentially expressed data set, in which preference will be given to differentially expressed data set irrespective of the entire human proteins. So in that case, you can use the differentially expressed data set as a background. Yeah, you can use gene set enrichment analysis also for calculating the enrichment of those genes. So means GACA itself is the another tool which you can use for the gene set enrichment analysis. Okay. So when we say expression, so it varies. So whenever we say protein expression, it means expression at the level of protein, not RNA expression. It is believed that if RNA expression is high, then its protein expression should also be high, but it is not the case, because there are some RNA which does not get converted into proteins. That's why what we have seen so far is that we cannot completely rely on RNA-seq information, because when we go to map the RNA-seq with the mass spectrometry data, which is specific for the proteins, so that does not align that much well. So when we say protein expression, we means at the level of proteins, at the level of peptides. So in protein expression, we will be focusing on the peptide or protein expression. And RNA expression level can act as a proxy to predict the protein expression, but it is not always the case. And one more thing which I want to add is that as we have seen that in mass spectrometry, because it is also having many challenges while interpreting. So in mass spectrometry, you're not means always able to find all the novel isoforms, or because when we say that there are some novel peptides which could be identified, what we mean by that is that means already experimental data sets or reference data sets such as UniProt which are present, so they're not that efficient to capture all the available protein sequences in the cell at current time, because mass spectrometry, because there are certain proteins which are like expressed for a very short interval of time. So in that case, what can happen is whenever you are collecting samples, that protein might be disease specific or that might be developmental stage specific. So in mass spectrometry, you might not be able to capture that sample. So in that case, you might not be able to find that protein. But by taking RNA-seq data, because it is based on the transcript level expression. So in that case, transcripts might be present. So you are more likely to get the unique protein sequences which might be available, but with mass spec you're not able to capture. So in certain scenarios, what people does is that they also take the RNA expression data to look into, means to comment about the protein studies, so protein expression studies. So that's why they take RNA expression levels in some studies. But when we say protein expression, so we are more likely focusing on the protein level expression. So what they have asked about FTU, did they ask about the variation of its expression in different organs? Okay, they have asked you about the highest expression in which organ. Okay, so what you can do is that, so from where you are getting this information of expression? Did you look it in the UniProt, this particular protein, from human protein at least? So means if you are not able to find there, then I think in gene cards you will be able to find the that value, because what I have mentioned is that in RNA-seq it is quite confusing. That's why in the this also I have mentioned it. So for example, this is the major tissues. So in this case, they are telling the RNA-seq level expression. That's why you will not be able to properly distinguish among different tissues. That's why I mentioned that if you go to gene cards, yeah, this is for FTO gene only. So in this case, they have mentioned it that it is expressed in the brain and specifically in the cerebral cortex. And in addition to it, it is also expressed in testes. So these are the mRNA expression in embryonic tissues and stem cells. And protein differential expression in normal tissues from HIPED for FTO gene, it is expressed in lymph node and retina. So you have to check which options are available there. So what I want to add from here is that you can use gene cards because it specifically will tell you in the end that which expression is more enriched, because they will calculate the enrichment score. So that with that they are telling it. Yeah, that might be the case. Or one more thing is that particular RNA is means specific to particular developmental stage or disease. So in that condition only it might appear. That is also one of the reason that you're not able to find the protein level expression. Yeah, means your question is true that It might have got transported or it might have got degraded. But transported and degraded terms comes when it has translated. But what if your RNA has not only translated? So there are multiple RNAs which could either be synthesized, but they are not required. So RNA can also get degraded instead of protein level degradation, and proteins can also get degraded. So these things might interfere with the protein expression levels. Yeah. Yeah, so means that only I want to add that when we say protein expression, we means expression at the level of protein, which is captured by the mass spectrometry or other immunohistochemistry analysis. But what is happening is that with means those assays or those mass spectrometry, we are not able to capture as many proteins. That's why we take mRNA expression as a proxy. But do not consider that mRNA expression is the protein expression. So they might not means align well with one another. So there are variations, and this is it. But in this case, as with mRNA expression only, we are taking these tissue specific information. So means by taking it as a proxy, that's why we are considering it. Okay, then we'll end this session and see you in the next session. Thank you so much. Today I've taken 32 more minutes. So thank you. I'll end it here.